So good afternoon, everyone, and welcome to the second PhasAGE Expert Seminar. This PhasAGE Expert Seminar series are a series of talks with invited speakers from the institutions that compose the PhasAGE Consortium, namely the I3S, the University of Padua, the Autonomous University of Barcelona, and the VIB Institute. There are 4 seminars planned for each year of the project. And these seminars will address the recent findings on PhasAGE scientific topics, including protein phase separation and protein aggregation in the context of aging and age-related disorders. And these seminars are open to the scientific community. So we hope that everyone enjoy the seminar, and I ask you to actively engage in the Q&A session at the end of the seminar. Feel free to write your questions on the chat, and Javier Garcia Pardo will moderate the seminar. So whenever you are ready, Javier, you can start to introduce the speaker. And thank you all for your attention and for attending this seminar.
Thank you, Rita, for your invitation and also for your nice introduction of this PhasAGE seminar. I will introduce, it's a pleasure for me to introduce the speaker today, which is Dr. Elsa Logarinho from the Universidad de Porto. And since 2015, she leads the Aging Aneuploidy Group, which is dedicated to study the mechanisms of the cell cycle, the regulation, and genomic instability, factors that contribute to aging and age-related diseases. And her research disclosed mainly chromosomal instability and as a hallmark of human aneuploid amniocytes. She published relevant papers in the last years in Life and high-impact journals. And currently, her group applies these interdisciplinary approaches to address how regulated instruction of proliferative fitness and genomic instability decelerates senescence in mouse models, including Down syndrome, progeria, and aging models. And within the A3S cancer program, Elsa is tackling the question of how aging is the major risk factor for tumor genesis, with a special focus on CAN and senescence. And she's currently the coordinator of the cancer program and member of the Restrictive Scientific Council. And she has been published over 35 publications with high-ranked journals in the field. And today she will present a recent work entitled Inhibition of Age-Associated Genomic Instability, emerging a strategy to delay cellular senescence in aging. And one of the major goals of aging research is to develop strategies to delay and even reverse aging. And emerging findings indicate that the genomic variations are relevant and unrecognized mechanisms underlying human aging and age-related diseases. And her group is studying this. And today she will present this important topic. Please, when you want, Elsa, you can start.
So I will share. So can you see my screen? Yeah?
Yes.
An innovation hub in the emerging field of biomolecular phase transitions in aging and age-related disorders. And I'm very thankful to Sandra Macedo Ribeiro for letting me join in. I must mention that I am not an expert in phase transitions, and this topic will not be covered in my seminar today, but I feel like I'm an expert in aging biology, in particular in different cellular and animal models of aging. And I wish that fruitful collaborations will arise in the context of the phage twinning. Since 2015, my group Aging and Unemployed at I3S in Porto has been studying genomic instability in the context of aging. And today's seminar is entitled Inhibition of Age-Associated Genomic Instability, an Emergent Strategy to Delay Cellular Senescence and Aging. So I will start by a brief introduction about aging. As you know, aging represents the major risk factor for many chronic diseases, including cardiovascular disease, diabetes, cancer, arthritis, and frailty. Aging occurs at the molecular, cellular, and organismal level. And a host of age-associated features have been identified, with a subset being potential drivers of the aging process. And these are called the cellular aging, the aging hallmarks. At the molecular level, aging hallmarks, they comprise DNA damage, epigenetic alterations, as shown here, also telomere attrition, and protein aggregation. At the cellular level, aging features include the accumulation of aberrant mitochondrial lysosomes, deregulated nutrient sensing, and cellular senescence. And senescent cells, they accumulate during aging and are the cells shown here in pink. The senescent cells, they are cells that stop dividing, and this is in response to stress or damage from exogenous or endogenous sources. And these cells, they exhibit distinct biomarkers, and they have a secretory phenotype that causes local chronic inflammation with an impact in the healthy neighbor cells. At the organismal level, the chronic low-grade inflammation leads to stem cells exhaustion and alter intercellular communication. In the lab, we have been focusing in chromatin aging, so in these primary hallmarks of aging, and how this leads to cellular senescence. Our rationale is that increased genomic and chromosomal instability during aging, they lead to a progressive loss of chromatin architecture inside the nucleus and epigenetic remodeling that in turn compromise the cell type specific gene expression that determines cellular identity and function. Although an intervention is still missing for the correction of DNA damage or otherwise to improve DNA repair mechanisms, other mainstream strategies are available that either improve the epigenome or target senescence. Aging, once considered irreversible, now is actually seen as remarkably malleable. The diet regimens such as caloric restriction and fasting, even more if coupled to circadian cycles, they are able to preserve the epigenome. More dramatically, cellular reprogramming by the Yamanaka effectors acts by resetting the epigenome, and this has been validated in animal models. But such a pluripotency triggering prevents its clinical translation thus far. So there is an intervention later on that is based on the ablation or clearance of senescent cells, and that has become increasingly popular. Senescent cells, they are responsible for the age-associated pathologies, and due to their secretory pro-inflammatory phenotype known as the SASP. And many drugs targeting senescence or the SASP, they are under clinical trials. One thing that caught our attention as we review in this paper in 2019 was the fact that when looking to reprogramming strategies or senolytic strategies, they all ended up by improving the cell proliferative fitness. So we ask whether by doing the other way around, so if we could somehow modulate the age-associated refractory features that we see in age cells, this could potentially work either to improve the epigenome or to delay senescence. To this end, we start by analyzing the cell cycle dynamics of age cells. We use human dermal fibroblasts that are retrieved from healthy Caucasian males with ages ranging from neonatal to octogenarian. And you can see here movies of early cell culture passages that were recorded under phase contrast microscopy for several days. When comparing a neonatal with a middle-aged sample, you can easily appreciate that a striking difference is the loss of proliferative capacity during aging. Even though it's widely accepted that this loss of proliferative capacity acts to prevent damaged cells from proliferating, the exact regulatory mechanisms have remained elusive. Also, we realized that the analysis of the cell division process in aging models and diseases was scarce, and the comprehensive analysis of cell division efficiency in natural age cells was largely missing. To address these questions, we established a human cellular model of natural aging that consists of human dermal fibroblasts retrieved from skin biopsies of healthy donors with ages ranging from neonatal to octogenarian. And since inter-individual difference exists in the rate at which a person ages, which is the so-called biological age, we included 2 donors of each age in a total of 5 chronological ages to increase the robustness of any correlation found with aging. In addition, we used dermal fibroblasts from an 8-year-old child with the Hutchinson-Gilford progeria syndrome as a model of premature cellular aging. Considering that during a normal postnatal lifespan, the cells in vivo will hardly reach the exhaustion number of replications that we observe in culture, and to limit any artifacts and clonal selection due to in vitro culturing, we only use early cell culture passages, which are way below the replicative lifespan exhaustion. To validate the suitability of our cellular models as models of normative and premature aging respectively, we measure the percentage of cells that exhibit senescence markers. And this chronic accumulation of macromolecular damage that occurs during aging, it induces a cellular stress response that we call senescence, and the accumulation of senescent cells occurs in age-related disorders. So the senescent cells, they have distinct biomarkers, such as increased beta-galactosidase activity, which is due to the increase in the lysosomal mass of the DDH cells. Also, heterochromatin foci, DNA damage markers, and the increased expression of cell cycle inhibitors, such as P16 or P21. In addition, these cells, they have a secretory phenotype of many bioactive molecules that are pro-inflammatory to the neighbor cells. And we measure senescent biomarkers in our cells, and we found that indeed there is an increase in the percentage of cells staining positive for beta-galactosidase, also staining double positive for a DNA damage marker and a cell cycle inhibitor marker, and they also increase in cell area with aging. So this validates the cellular model as representative of natural aging and progeria. To investigate the cell cycle behavior of fibroblasts from different age individuals, we performed long-term phase contrast live cell imaging. And interestingly, we found that not only the number of cells that divide decreases with increasing age, but also the interval between the nuclear envelope breakdown and the anaphase onset, which we use this interval to measure the mitotic duration, increases with advancing age. So mitosis becomes delayed as we approach later ages. And this is a steady increase in the mitotic duration. To investigate the chromosome or spindle, microtubule spindle defects that contribute to this increased mitotic duration in the older mitotic cells, we perform high-resolution spinning disk confocal microscopy in cells that were transduced with lentivirus for the expression of H2B-GFP to label the chromosomes and M-Sherry tubulin to label microtubules on the mitotic spindle. And we found that there is an increase in several mitotic defects with aging, significantly from middle age on. And these include chromosome misalignment or delay in the congression of chromosomes to the metaphase plate, and also an increase in the number of missegregation events, such as the one shown here with the anaphase lagging chromosome. In agreement with these mitotic defects, we also found that there is an increase in aneuploidy. And we measure aneuploidy by doing fluorescence in situ hybridization analysis, both in a synchronous cell and post-mitotic cells. And post-mitotic cells here means cells that have just gone through a mitotic division. So these are telophase cells. And you see that from middle age on, aneuploidy increases. It is also increasing in aneuploidy in progeria samples. And the same, it's even more striking if we look to cells that have just divided. So aneuploidy increase during normative aging, and this is due to an age-associated loss of mitotic fidelity. To identify the molecular mechanisms behind this complex aging-associated mitotic phenotype, we perform RNA sequencing gene expression profiling of cells captured in mitosis accordingly to the experimental layout shown here. So basically, we pick at neonatal cells and elderly cells, and we block them in mitosis using a mitotic drug that we call STLC. And this treatment is done overnight. So this allows to enrich the mitotic index of the cultures. And then we collect mitotic cells by doing a mechanical detachment. And we use these mitotic cells for RNA-seq analysis. You see here that the populations that we got through this procedure, they are highly enriched for mitosis, like 95% of efficiency. And so using these mitotic cells, we performed the RNA-seq profiling, and we found that in the top 10 geoterms, the most altered genes were cell cycle and mitotic genes. When comparing the geoterm for mitotic genes, we actually saw that comparing with neonatal cells that had highly expressed mitotic genes, the elderly cells, they have an overall downregulation of these genes. And it seems to be quite a global shutdown for most of mitotic genes. Interestingly, these genes have been reported as targets of the MMB FOXM1 transcriptional complex, which drives the expression of late cell cycle genes that contain in their promoters this homologous region CHR. Therefore, we ask whether during aging, there was a loss of mitotic proficiency due to FOXM1 downregulation, which is a component of the MMB complex. Indeed, increasing age correlated with decreased FOXM1 transcript and protein levels in our human samples. We thus concluded that an age-associated FOXM1 repression accounts for an extensive transcriptional downregulation of mitotic genes and the phenotypes that we observed in the early senescent dividing cells. Moreover, a comprehensive list of 55 genes identified as comprising the universal senescence signature was interrogated in our RNA-seq data. And although the transcriptome of senescent cells is highly heterogeneous, depending on the cell type or the senescent stimulus, these 55 genes were identified as being conserved in all types of senescent cells. And looking to this signature, we found that in our RNA-seq data set of the mitotic elderly fibroblasts, 19 genes of this signature were altered, and 15 of these genes were behaving as expected, because there are some genes that should be downregulated in senescent cells, as it is the case here, and others should be upregulated, as shown here. So an unforeseen SAS phenotype seems to evolve in elderly dividing cells alongside a global transcriptional shutdown of mitotic genes, accounting for the age-associated mild aneuploidy levels. To test the correlation between FOXM1 repression and age-associated phenotypes further, we depleted FOXM1 in fibroblasts from a young donor and using RNA interference. Again, we did the RNA-seq profiling of the SI-FOXM1 depleted cells using mitotic cell populations. And we found that, interestingly, 50% of the genes that are downregulated or altered in FOXM1 RNAi interference, they are also altered in elderly fibroblasts. So 50% of the genes altered during aging seems to depend on FOXM1 expression levels. Again, the major geoterms that we found are geoterms related with cell cycle regulation and mitosis. In agreement with what we have previously seen for the elderly fibroblasts, we saw that the RNA interference of FOXM1 also induced mitotic delay. It also increased the incidence of aneuploidy, and there was also an increase in the percentage of cells staining positive for senescent markers, and also the genes associated with the senescence secretory phenotype were being upregulated, as shown for the elderly fibroblasts. So these results show that in young cells with low levels of FOXM1, equivalent to those that we had in the octogenarian cells, they recapitulate the aging-associated mitotic defects, aneuploidy, and senescent phenotypes. So we then did the obvious question: are age-associated phenotypes rescued by FOXM1 induction? For this, we use the expression of a constitutively active truncated form of FOXM1. And this is an N-terminal deleted isoform because the N-terminal has an auto-inhibition regulatory loop that inactivates the transactivation domain. We lentivirally infected octogenarian fibroblasts with this truncated form that you see here. And again, we did the RNA-seq profiling to see the gene expression signature of these octogenarian cells with constitutively active FOXM1. We found that there is an increase in the abundance of 80 mitotic cell cycle genes, and that most of the genes overlapped extensively with those that were altered in the elderly fibroblasts and also in the FOXM1 RNAi. And in agreement the age-associated mitotic defects that we saw previously in the elderly fibroblasts, they were rescued upon induction of FOXM1 activity. And this also led to a decrease in the incidence of missegregation events and aneuploidy. FOXM1 overexpression also decreased the percentage of cells with senescent biomarkers, and consistently also to the amelioration of the SASP and senescent core transcriptional signatures. So overall, this data demonstrate that modulation of mitotic efficiency through FOXM1 induction in elderly cells prevents aneuploidy and delays cellular senescence. But then we ask, why does modulation of mitotic fitness delay cellular senescence? We thought that this could be related to the missegregation events. And to directly demonstrate that were the chromosome missegregation events that were acting as ultimate trigger to the establishment of a full senescent phenotype, we performed the experimental layout shown here. Octogenarian fibroblasts expressing H2B GFP, they were followed under long-term time-lapse microscopy. We use a plate with coordinates, and at the end of the movie, so we record during 3 days, at the end of the movie, we fix the cells, we stain for senescent biomarkers, and then following the plate coordinates under the same microscope, we can correlate the fixed cell with the live cell analysis. And dotted cell fate of the mitotic cells that we saw dividing on the first day was a measure for cell death, senescence onset, or cell cycle. Doing this experiment, what we found was that when the mother cells divide correctly in the first day, the daughter cells of these mother cells, they will most kept on cycling, and only a very few arrest the cell cycle, and only 7% of these ones that arrest the cell cycle became positive for senescent biomarkers. However, when doing the same experiment but following those cells that divide with chromosome missegregation events, and here you see 2 daughter cells that have micronuclei coming from this chromosome missegregation during mitosis. If you look to this daughter cell, you will see that by the end of the movie, we fix the cells, we stain for the senescent biomarkers, and you see that this micronucleated daughter cell became positive for the senescence beta-galactosidase activity, and also the DNA damage and cell cycle marker. So the conclusion of this experiment was that whenever the mother cell divides with missegregation events, it generates daughter cells that always stop from cycling, so they become arrest in the cell cycle, and 85% of the cells, we found that they become senescent. So the conclusion from this study that was published in this Nature Communication paper was the following. Basically, we found that proliferating naturally aged cells, they have FOXM1 repression, and they have also a senescence-associated transcriptional priming that we call early senescence, that leads to a loss in mitotic fidelity, chromosome missegregation, that ultimately triggers an accumulation of full senescent cells in the cell culture. These senescent cells, of course, they send paracrine inflammatory signaling to the neighbor cells, also compromising their proliferation fitness. By reinstating FOXM1 transcriptional activity in the elderly cells, we saw that this ameliorates not only cell autonomous, but also cell non-autonomous feedback effects between the mitotic fidelity and senescence, suggesting that this mechanism evolved as a positive feedback between cellular aging and aneuploidy. If chromosome segregation then accounts for full senescence, this means that if we could somehow inhibit chromosomal instability, this should work to delay senescence. Chromosome segregation, as I told you, is often due to these lagging chromosomes, as one that you see here in the movie. And lagging chromosomes, they originate whenever overstable erroneous interactions between the chromosomes and the microtubules are left incorrected into anaphase. The stability of kinetochore microtubule interactions, it's tightly regulated, and it must be within levels that are enough stable to allow the formation of the attachments, but also enough unstable to allow the correction of erroneous attachments. And this error correction machinery is safeguarded by the activity of two protein motors, the microtubule destabilizing kinesin 13 proteins KIF2B and MCAC. So we ask if the microtubule, if the kinetochore microtubule dynamics was being affected in the elderly cells. And indeed, we saw that there are reduced MCAC levels in elderly cells, as shown here by Western blot analysis and also immunofluorescence analysis of mitotic cells. And as expected for reduced MCAC levels, we found that there is an increased stability of what we call the K fibers, which are these microtubules that bind to the chromosome kinetochores. And you see that this overstability of the microtubules is specific at the metaphase stage of mitosis, and its increase in the elderly cells. So we went on testing an agonist of the microtubule depolymerizing kinesin MCAC, known as UMK57, in the elderly cells. This agonist had been previously published by Dwayne Compton Lab as a strategy to tackle chromosomal instability in cancer cells. And so to see whether this agonist could bring any benefits to the microtubule overstability seen in the elderly cells, we start by titrating the optimal dose concentration that would work to decrease the microtubule overstability in the elderly cells. We found that at 1 micromolar concentration, we could rescue the mitotic delay that we saw for the elderly fibroblasts. And importantly, using this concentration, there was no impact in the young neonatal fibroblasts. Indeed, using 1 micromolar treatment, the overstable microtubule attachments were rescued. So now the microtubule density is very similar to the density that we saw in the young fibroblasts. And as expected, this translated into a lower chromosome missegregation rate. So these are the elderly cells treated with UMK57. And also this prevented the generation of micronuclei that were staining positive for cGAS. And this is important because the cGAS STING pathway is a pathway that when active leads to the onset of senescence, because it's the pathway that detects cytosolic DNA. And because all this is being reduced, we also saw that the senescence accumulation was being delayed in the elderly cells treated with this drug. So in conclusion, we disclose the first small molecule inhibition of chromosomal instability that delays senescence. We found that disturbed kinetochore microtubule interactions are a major driver of chromosome instability, which increases with aging and contributes to senescence. The aged cells, they display the effective microtubule depolymerizing kinase insertin activity and kinetochore microtubule error correction. And this chromosome segregation fidelity could be restored by the small molecule enhancement of MCAC activity. And this destabilization of the kinetochore microtubule interactions could help to delay the cellular aging phenotypes that were associated with chromosomal instability, micronucleation, cGAS-STING activation, and senescence onset. This work has been published in at Tembo Reports in 2020. So we are now addressing the impact of both FOXM1 genetic modulation and the MCAC agonist pharmacological treatment in all the other cellular aging hallmarks, such as the epigenome, the nuclear architecture, and the DNA repair mechanisms. And we are also investigating if there are benefits at the organismal aging. We hope to disclose these findings in a very near opportunity. So I finish with this slide just to highlight that we have cellular and animal models available to address phase transitions topic. I believe that phase transitions, they are an hallmark in aging and aneuploidy, in the sense that they can drive aging and their modulation could work to prevent these pathologies. The closest we have been into the topic was in this previous study that we published in 2019, where we demonstrated that both the morphology and the function of the nucleolar compartment changed during aging and actually works as an aging biomarker. So I finish by acknowledging our funding agencies and the members in the lab, particularly Joana Macedo, Mónica Vilares, Rui Ribeiro, Sara Vaz, who contributed to the work that I've shown here today. Our collaborators, Dwayne Compton at Dartmouth, USA, and his lab members that help us in the study with the MK57 compound. And also in Netherlands, Floris Voiger and René Medema, who supported our work with the FOXM1 modulation. Thank you for your attention, and I'm pleased to take any questions.